Chemiosmotic theory explains the intricate relationship among electron transfer, electrochemical gradient formation, and ATP synthesis. Oxidative ATP synthesis in mitochondria is dependent on electron transfer through an electron transport chain consisting of multi-protein complexes arranged with progressively high standard reduction potential. Therefore, Mitchell's chemiosmotic theory explains enzymatic reaction involving simultaneously occurring transport process and chemical reaction, indicating strongly that both processes are intricately coupled. This coupling has been experimentally proven as described below. When oxidizable substrate like succinate is added to a buffer-containing mitochondria, ADP and pi, the following processes commence. The substrate is oxidized, O2 is consumed, which is monitored by an oxygen electrode, and ATP is synthesized. Since energy liberated following substrate oxidation drive ATP synthesis by transferring an electron to O2, therefore, by use of electron transfer inhibitors such as antimycin or cyanide, ATP synthesis would be blocked. More surprisingly, the opposite was also found to be true, i.e., blockage of ATP synthesis inhibits electron transfer. The obligatory nature of coupling in mitochondria can be shown by providing succinate and O2, but no ADP and pi. Under these circumstances, neither ATP synthesis nor electron transfer to O2 occurs. Coupling of oxidation, electron transfer to O2, and phosphorylation ATP synthesis can also be demonstrated by the use of oligomycin or venturicide. Although oligomycin inhibits ATP synthesis without interfering with the electron flow, but later is also affected, indicating the dependence of one process on the other. Figure 1.66 electron transfer and ATP synthesis are coupled in mitochondria. In this experimental setup, mitochondria are suspended in a buffer solution containing ADP and pi. Level of consumption of O2 is monitored by an oxygen electrode. Addition of ADP and pi does neither increase the O2 consumption nor lead to ATP synthesis. But the addition of oxidizable substrates such as succinate increases both O2 consumption and ATP synthesis, which are inhibited by cyanide, an inhibitor blocking electron transfer between cytochrome oxidase and O2. Wheat mitochondria provided with succinate respires and synthesized ATP only after the addition of ADP and pi. Subsequent addition of ATP synthase inhibitors such as oligomycin or venturicide inhibits O2 consumption, respiration, and ATP synthesis. Dinitrophonyl acts as an uncoupler, inhibiting ATP synthesis without affecting respiration. ATP synthase is also popularly known by several names such as proton translocating ATP synthase, F1FO ATPase, complex B, and F-type H plus ATPase, molecular motor, etc. This complex utilizes energy conserved in electrochemical gradient proton motive forcing order to bring about ATP synthesis. Ephraim Rapper discovered that ATP synthase consists of two distinct functional units designated as F1, a peripheral water-soluble protein, and FO means oligomycin sensitivity, an integral water-insoluble membrane protein. During experiment, if the outer mitochondrial membrane is removed and the inner mitochondrial membrane is sonacated, it results into the formation of numerous sealed, almost spherical shaped vesicles called submitochondrial particles. These particles contain ATP synthase in outward orientation and are capable of ATP synthesis. However, if these particles are stripped of the F1 subunit by urea treatment, it loses its ATP synthesizing activity, which is restored upon reconstitution, as shown below. F1 consists of 5 different types of subunits, which can be easily separated from FO following treatment of submitochondrial particles with urea. It has been experimentally proven that solubilized F1 loses its ATP synthase activity but gains ATP hydrolase activity, hence the name ATPase. Mixing of F1 with FO-containing mitochondrial particle restores its ATP synthase activity. Moreover, several bacterial F1 FO acids translocate not plus ions rather than H plus. Figure 1.67 formation of submitochondrial particle. Ultrasonic disruption of the inner mitochondrial membrane following removal of the outer membrane results into the formation of submitochondrial particles containing outwardly projecting F1 particles and possess the capability of ATP synthesis under appropriate conditions. Treatment of submitochondrial particles with urea results into stripping of membrane and separation of F1 particles with loss of ATP synthesizing activity. Mitochondrial ATP synthase contains F1 and FO subunits. Each F1 consists of 9 subunits belonging to 5 different types, with a composition E3B3 grams. Of this no namer, each V subunit contains a catalytic site for ATP synthesis. The A and B subunits of F1 are arranged alternately, each with a specific conformation. At any given point of time, each V subunit has different conformations. For example, one B subunit has conformation appropriate to ATP binding, designated BTP or BATP. The second B subunit's conformation could be appropriate for ADP plus pi binding, designated BTP or BADP, whereas the third B subunit is empty, designated B or BMP. This was proven by crystallizing F1 protein in the presence of AP-NHP, a non-hydrolyzable structural analog of ATP and ADP. After analysis, it was found that one B subunit contained AP-NHP, second B subunit was bound with ADP, while the third one was empty. Besides, a subunits also bind ATP and show conformational differences from each other. Each N B subunit contains water A and water B motifs with specific amino acid sequences, which participate in ATP binding. These conformational differences among N B subunits, along with the presence of G subunits, make F1 asymmetric protein. The FO complex contains 3 subunits designated as A, B, and C, with composition F2C10 to 12. The number of C subunits is highly variable, ranging from 10 to 12 as well as it is very hydrophobic. Each C subunit contains 2 helices. Figure 1.68 mitochondrial ATP synthase complex. This diagram of F1 FO ATP synthase is based on crystallographic and biochemical studies. Each F1 is no namer with a 3B3 grams of composition, whereas FO has F2C10 to 12. 2 B subunits of FO remain associated with F1s and B subunits. The C subunits C10 to 12 make the integral membrane protein cylinder which remains attached to the shaft of F1 made up of G and epsilon. The flow of protons through FO rotates the cylinder and shaft. This rotation brings about a conformational change in B subunits as and when they come in contact with G subunits one after another. B AP-NHP, a non-hydrolyzable structural analog of ATP. Binding change mechanism of ATP synthesis. Paul Boyer proposed a mechanism of rotational catalysis underlying ATP synthesis based on insights developed from kinetic and binding studies of reactions catalyzed by ATP synthase. The 3 active B subunits sequentially undergo differential conformational change about each other into the relative orientation of other proteins, such as G, in the complex. The conformational change, brought about due to rotation, is induced by conserved free energy and protons streaming through the FO portion of ATP synthase. The 3 B subunits at any given point of time have differential. Figure 1.69 rotational catalysis of ATP synthesis. Conformation, of which one is specific for ATP plus pi binding, one for ATP, and the remaining is empty, and accordingly, they are designated by BADP or L, loose site BATPT, tight site and BMTO, open site respectively. The phosphone hydride bond between B phosphate of ADP and pi is formed only in the T state, whereas newly synthesized ATP is released from the O state. The B subunit with L state binds to free ADP and pi from the surrounding medium. Thereafter, the ADP pi bound L site undergoes free energy driven conformational change to T site, in which the formation of phosphone hydride bonds is induced, forming ATP. There are differential conformations among 3B catalytic sites, which is determined by the relative position to the rotating G subunit. At any given moment, the catalytic site of one B subunit is compatible to binding with ADP plus pi BADP site or L state, one is compatible with ATP binding BATP site or T state, and the third one is in the open conformation B empty site or O state. The rotation of epsilon GC10 to 12 assembly, caused by proton motive force, brings G subunit in transient contact with each F subunit pair in succession, inducing conformational changes and thereby release of newly synthesized ATP. Inhibitors of oxidative phosphorylation are several inhibitors, either derived from plants or chemically synthesized, that are known to inhibit oxidative phosphorylation at multiple stages. These inhibitors have been proven to be very useful in deciphering the order of complexes of the electron transport chain and its functioning, as stated in the earlier part of the chapter. There are 4 complexes in the electron transport chain and ATP synthase, also called complex B, albumin and direct part of the chain, which are specifically inhibited by specific inhibitors. Figure 1.70 inhibitors of electron transport chain. These inhibitors inhibit electron transport and thereby stop ATP synthesis. Shuttle systems of cytosolic NADH into electron transport. TCA cycle generates most of NADH in the mitochondrial matrix, which is quickly oxidized through the electron transport chain. However, NADH generated by glycolysis cannot transfer its electrons to and get oxidized by the electron transport chain owing to the impermeability of the inner membrane for NADH. Therefore, 2 different shuttle systems have evolved to complete this task. Malate aspartate shuttle. This special system is functional in the heart, kidney, and liver. The basic underlying mechanism is that the cytosolic NADH reduces mitochondrial NAD+ through the intermediate reduction and subsequent regeneration of oxaloacetate. The reducing equivalence of cytosolic NADH is first transferred to cytosolic oxaloacetate, thereby forming malate and NAD+. This reaction is catalyzed by cytosolic malate dehydrogenase. The malate, thus formed in the cytosol, is transported to the mitochondrial matrix in exchange for A-ketoglutarate from the matrix by malate A-ketoglutarate carrier. Once within the matrix, mitochondrial malate dehydrogenase oxidizes malate to oxaloacetate to yield NADH from NAD+. Therefore, one cytosolic equivalent NADH is formed in the matrix. This NADH can pass a pair of electrons directly to O2 through the electron transport chain, generating 2.5 ATP. Glycerol 3-phosphate shuttle. It occurs in skeletal muscle and brain, and is particularly prominent in insect flight muscle. This shuttle is simpler but has less energy as it generates only 1.5 ATP for every cytosolic NADH. This shuttle system involves the enzyme glycerol 3-phosphate dehydrogenase, which catalyzes the reduction of dihydroxyacetone phosphate to glycerol 3-phosphate with simultaneous oxidation of NADH to NAD+. This NAD+ is again used in glycolysis and other reactions occurring in the cytosol. The glycerol 3-phosphate transfers its electron to glycoprotein dehydrogenase, located on the outer face of the inner mitochondrial membrane, to yield FOD2, which eventually transfers electrons to complex 3-beta ubiquinone. Therefore, it yields only 1.5 ATP. Figure 1.71 mechanism of malate aspartate and glycerol 3-phosphate shuttle. This shuttle system helps in carrying reducing equivalents from cytosolic NADH to mitochondrial matrix and yields 2.5 ATP. Whereas glycerol 3-phosphate shuttle is an alternative way of conveying cytosolic reducing equivalents to the matrix and yields 1.5 ATP. Enzyme. Enzyme classification. Enzymes have been named by a joining suffix ase to their substrate name or to the word defining their catalytic activity. For example, DNA polymerase is known to catalyze the polymerization of nucleotides to form DNA, and urease is known to catalyze the hydrolysis of urea to ammonia and carbon dioxide. For a precise identification of all the enzymes, the International Union of Biochemistry set up an enzyme commission in 1964 to develop a systematic nomenclature for enzymes. Depending upon the nature of the chemical reaction catalyzed by them, enzymes have been classified into 6 major classes. Table 1.10. Class type of reaction example. 1. Oxidoreductase is oxidation reduction, transfer of electrons. Lactate dehydrogenase. 2. Transferase is group transfer reactions nucleoside monophosphate kinase. NMP kinase. 3. Hydrolase is hydrolysis reactions chymotrypsin. 4. Lyase is removal of chemical groups from the substrate leaving a double bond. Fumarase. 5. Isomerase is isomerization reactions triphosphate isomerase. 6. Ligase is joining of two substrates coupled to ATP hydrolysis. Amino acyl synthetase. Table 1.106 major classes of enzymes. Enzyme is a catalyst. A large number of biochemical reactions comprising life are mediated by a series of extraordinary and potent proteins, the enzymes. Enzymes are biocatalysts possessing remarkable catalytic ability and high specificity for the substrates. Enzymes catalyze the synthesis of biological macromolecules from their precursors, breakdown of nutrient molecules, and conversion of chemical energy. However, the action of biological catalysts differs from ordinary synthetic or inorganic catalysts in the following aspects. 1. Higher reaction rates. The rate of reactions catalyzed by enzymes tends to be 10 6 to 10 12 times higher compared to the uncatalyzed reactions. For example, the breakdown of hydrogen peroxide into water and oxygen catalyzed by catalase is 10 times faster as compared to the uncatalyzed reaction. 2. Mild reaction conditions. Generally, an increase in temperature and pressure leads to increased reaction rates in chemical catalysis. But enzymes are known to function efficiently in mild temperature and pH conditions. 3. Higher specificity. Enzymes possess huge specificity for the substrates and the nature of the reaction to be catalyzed by them. The enzyme catalyzed reactions rarely lead to the formation of any wasteful byproduct through side reactions. 4. Capacity to regulate. The rate of metabolic sequence in cells can be regulated by controlling certain enzyme activities. These regulatory processes include covalent modification of enzymes, allosteric control and alterations in the number of enzymes synthesized. Enzyme rate enhancement cyclophilin 10 5 triisomate mutase 10 6 carbonic anhydrase 10 7 triphosphate isomerase 10 9 carboxypeptidase A10 1 1 amp nucleosidase 10 12 6 cotransferase 10 13 urase 10 140 M PD carboxylase 10 17. Table 1.11 catalytic power of some enzymes. Enzymes, like any other chemical catalyst, take part in the reaction and reduce activation energy by providing an alternate pathway to the reaction, thereby accelerating them. The enzymes do not go through any permanent change and can be retrieved as such at the reaction completion. Most of the enzymes known are proteins except a group of RNA molecules, and the three-dimensional structure presents an active site for binding the substrate molecules to their surface. The catalytic action of many enzymes is dependent upon the existence of additional chemical components known as cofactors. Table 1.12. Apoenzyme inactive plus cofactor holoenzyme active. Cofactor enzyme coenzyme. Biotin pyruvate carboxylase thymine pyruphosphate pyruvate dehydrogenase flavin adenine nucleotide monoamine oxidase pyridoxal phosphate glycogen phosphorylase tetrahydrofolate thymolate synthase coenzyme A acetyl carboxylase nicotinamide adenine dinucleotide lactate dehydrogenase cofactor enzyme. Metal. K plus pyruvate kinase Ne2 plus urate. Say glutathione peroxidase Mn2 plus superoxide dismutase non-nitrate reductase. Fe2 plus or Fe3 plus cytochrome oxidase catalase peroxidase Mg2 plus hexokinase glucose 6 phosphatase. Pyruvate kinase Cn2 plus carbonic anhydrase carboxypeptidase A and B alcohol dehydrogenase. Table 1.12 enzyme cofactors. How do enzymes work? Enzymes are an indispensable part of every biochemical process. They accelerate the reactions by a factor of millions, and in their absence, most of the cellular reactions will not take place at all, even over a period of years. Hence, they are essential to sustain life. Enzymes extracted from the cells can be used to catalyze important processes, such as the production of cleaning products to antibiotic modification. The enzyme catalyzed reactions take place in a specialized region of the protein called an active site. Free energy. The free energy change (DG) of reaction is the difference between the free energy of products and the free energy of the reactants. It is quite different from the energy required to begin the conversion of reactants into the products. The free energy change between the products and the reactants can predict the thermodynamic feasibility of a reaction. A negative DG value signifies that the reaction is spontaneous in nature and is called an exergonic reaction. If DG is 0, the system is said to be at equilibrium and no net change takes place. A reaction having a positive DG value cannot take place spontaneously and is said to be endergonic in nature. Though a negative DG value tells about the spontaneity of the reaction, it sheds no light on how fast it is. Figure 1.72 reaction coordinate diagram. Reaction coordinate. We can write a simple enzyme catalyzed reaction as E plus S rightwards harpoon over leftwards harpoon S rightwards harpoon over leftwards harpoon E P rightwards harpoon over leftwards harpoon E plus P. Where E, S, and P represent the enzyme, substrate, and product. S and E P are complexes of the enzyme with the substrate and with the product. The reactants approach each other along the pathway of minimum free energy called reaction coordinate. When the free energy of the system is plotted against the reaction coordinate, it is called a reaction coordinate diagram. In this figure, the reactants and the products represent the state of minimum free energy, and the transition state corresponds to the highest free energy in the figure. The reaction SP can be illustrated by the reaction coordinate diagram. Binding energy. The enzymes possess an extraordinary ability to catalyze the biochemical reactions by dramatic lowering of the activation energy. Till now, we have understood the fact that enzymes enhance the rate of the reaction by promoting the formation of the transition state and decreasing the activation energy. The enzymes align the substrates in favorable arrangements, facilitating the formation of the transition state in enzyme substrate S complexes. The formation of enzyme substrate complex through weak forces of interaction like hydrogen bonds, hydrophobic and ionic interactions helps in stabilizing the protein structure. The formation of enzyme substrate complex by these weak non-covalent interactions results in the evolution of free energy, which helps in stabilizing the interaction to a large extent. The energy evolved from the formation of the enzyme substrate complex is known as binding energy and is significantly involved in reducing the activation energy of the reaction. The diagram describes energy changes taking place during the reaction progress. The enzymes speed up the reaction by decreasing the activation energies. DG does not equal. The free energy change for the reaction is designated by DG. Activation energy. As discussed, the free energy change of reaction gives an idea regarding the equilibrium of the reaction, and in the presence of enzymes, this equilibrium is attained quickly. To figure out how enzymes enhance the rate of the reactions, we have to consider the pathway leading to the conversion of reactants into the products. All the biochemical reactions possess an energy barrier which prevents the reaction to proceed spontaneously. Some energy is always required by the reactants for the arrangement of the reacting groups, bond rearrangements, creation of temporary unstable charges, and other conversions before they are transformed into the products. This input of energy required to start the reaction or break the barrier is called the activation energy. Transition state. During a chemical reaction, the molecular structure of the reactant transforms into the structure of the product. In this process, some bonds are broken and some are formed. The highest energy arrangement of atoms, which is intermediate in structure between the structure of reactants and the structure of products, is known as the transition state. A chemical reaction involving the transformation of substrate S into product P occurs via the formation of transition state S having higher free energy compared to S and PO into the highest free energy of transition state. It is rarely occupied species in the course of the reaction. It is important to mention here that the transition state is quite different from a reaction intermediate as it is not a chemical species with any significant stability. The difference in the energy levels of the ground state and the transition state is the activation energy. Entropy reduction. Although the binding energy accounts for a significant reduction in activation energy, there are other thermodynamic and physical factors contributing to the reaction barrier which need to be considered. These factors include lowering of entropy due to restrained freedom of motion of molecules in solution B, the solvation shell of water surrounding and stabilizing biomolecules in an aqueous medium C, the required deformation of substrate molecules for reaction to take place D, the requirement for precise arrangement of catalytic residues on the enzyme. Mechanism of enzyme action. The mechanism for enzyme action can be seen as comprising of three events. First of all, the enzyme binds to the substrate forming an enzyme-substrate complex which ultimately gives the product. These stages in enzyme catalyzed reaction can be represented as E plus S rightwards harpoon over leftwards harpoon S rightwards harpoon over leftwards harpoon E P rightwards harpoon over leftwards harpoon E plus P. Where E is the enzyme, S is the substrate, and S is enzyme-substrate complex and P is the product. It has been well established that the enzyme steps up the reaction by the reduction in activation energy and stabilizing of a transition state. The enzyme brings the substrate together in the most favorable orientation and aids in the development of a transition state in enzyme-substrate complex. The enzymes exhibit selectivity while binding a substrate and can easily discriminate between the substrate and similar molecules. Different theories have been proposed to explain the formation of S complex by the interaction of enzyme and substrate. Lock and key model. In light of the substrate specificity exhibited by the enzymes, Emil Fischer in 9184 proposed lock and key hypothesis to explain the binding between the substrate and the enzyme. The basis of this hypothesis is the complementarity between the enzyme and the substrate, figure 1.73. It suggested the fitting of substrate. Reaction rate and equilibrium. To understand the working of the enzyme, the thermodynamic relationship of reaction equilibrium to the standard free energy change for reaction, dg, degree and the rate of reaction to the activation energy, dg degree, needs to be introduced. The standard equilibrium constant, K, EQ for reaction S rightwards harpoon over leftwards harpoon P, is defined as EQ, P, K, S, equals 1. A negative dg, degree value represents favorable reaction equilibrium, but it provides no information regarding the rate of the reaction. The rate of reaction is represented by the product of reactive concentration and by rate constant K. For a unimolecular reaction S rightwards harpoon over leftwards harpoon P, the rate of expression is given by the expression V equals KS3. This is the expression for first order reaction as the rate depends upon the concentration of S only. K is the proportionality constant which tells about the feasibility of the reaction under given conditions. If the rate of reaction is dependent upon two different compounds, the reaction is second order and the rate equation for such reaction becomes V equals KS1 S2. The expression relating the rate constant and activation energy can be derived from the transition state theory. G R T K T K E H minus increment equals 4, where H is the Planck's constant, K is the Boltzmann constant. Since the relation between the activation energy dg and the rate constant K is inverse and exponential, it proves that the lesser the activation energy, the reaction will proceed at a rapid rate. Product. Although this hypothesis was able to explain why an enzyme only works on certain substrates, it is unable to explain the mechanism of the enzyme. Molecule into the enzyme's active site similar to a key fitting into the lock. It leads to the formation of transient S complexes, which ultimately break down into the enzyme N. Figure 1.73 lock and key model. The lock and key model suggests that the substrate is complementary to the active site of the enzyme. Induced fit model. Daniel E. Koshland put forward the concept of flexibility of enzymes and suggested that the shape of the active site is transformed by the substrate binding. Hence, Koshland postulated the induced fit hypothesis, which stated that enzyme structure is disparate depending upon either substrate is bound or not. Upon substrate binding, the enzyme is subjected to conformational modification, which is responsible for aligning the specific functional groups into the proper orientation for speeding up the reaction. Figure 1.74. This conformational change also justifies the onset of increased weak bonding interactions in the transition state. Figure 1.74 induced fit model. In this model, the enzyme undergoes conformational change on substrate binding and then forms a shape complementary to the substrate. Different catalytic processes of enzymes. The first step of catalysis by an enzyme is substrate binding. This binding mediated by weak interactions between the substrate and the enzyme release. Induced fit model. Daniel E. Koshland put forward the concept of flexibility of enzymes and suggested that the shape of the active site is transformed by the substrate binding. Hence, Koshland postulated the induced fit hypothesis, which stated that enzyme structure is disparate depending upon either substrate is bound or not. Upon substrate binding, the enzyme is subjected to conformational modification, which is responsible for aligning the specific functional groups into the proper orientation for speeding up the reaction. Figure 1.74. This conformational change also justifies the onset of increased weak bonding interactions in the transition state. Figure 1.74 induced fit model. In this model, the enzyme undergoes conformational change on substrate binding and then forms a shape complementary to the substrate. Different catalytic processes of enzymes. The first step of catalysis by an enzyme is substrate binding. This binding mediated by weak interactions between the substrate and the enzyme releases the free energy, called the binding energy, responsible for increasing the catalytic efficiency of the enzyme. The substrate binds to the enzyme in such a way that the arrangement of the catalytic groups aids in breaking and creating bonds by several mechanisms such as general acid-base catalysis, covalent catalysis, and metal ion catalysis. Acid-base catalysis. In general acid-base catalysis, the rate of reaction is enhanced by proton transfers from molecules other than water. The metabolic pathways lead to the generation of intermediates that are highly unstable and tend to break down into reactants, hence preventing the reaction to occur. The stabilization of these charged intermediates is done by proton transfer from these intermediates to give a species that can be converted into a product. An uncatalyzed keto-enol polymerization reaction takes place at a slower rate due to the formation of an unstable carbonyl-like transition state. In catalyzed reaction, the carbonyl character can be decreased by donating a proton to the oxygen atom, thereby enhancing the reaction rate. The active site of the enzyme consists of a large number of amino acid side chains asp, blue, his, CYs, tier, and Li, which can act as proton donors and acceptors because they have pK's value near physiological pH. The precise location of these groups in the active site of enzymes allows the proton transfers providing the rate acceleration in order of 10 to the 10 5. Metal ion catalysis. Metal ions take part in catalysis through various ways. Depending upon the strength of ion-protein interaction, metal ion requiring enzymes can be classified into two groups. 1. Metal enzymes consist of metal ions that are bound tightly like Fe2+, Cu2+, Zn2+, Mn2+, Co3+. 2. Metal activated enzymes pick up loosely held metal ions from a solution along with substrate like Na+, K+, Mg2+, Ca2+. Such type of ions is generally involved in playing a structural role instead of a catalytic role in the enzyme. Metal ions take part in the catalytic process in three major ways. 1. Metal ions possess the ability to stabilize a reaction intermediate carrying a negative charge by serving as an electrophilic catalyst. 2. The interplay of ionic interactions when substrate binds to enzyme bound metal aids in orienting them properly for reaction. 3. The reversible changes in the metal ion oxidation state enables them to carry out oxidation reduction reactions. In the metal ion catalyzed reactions, the metal ions usually help in neutralizing a negative charge similar to a proton. Yet, the metal ion is far more effective as compared to proton due to the presence of a higher concentration of metal ions at physiological pH and may possess a charge higher than +1. The positively charged ion, when binds to water molecules, leads to a lowering in the pKa of water, hence generating a substantial concentration of nucleophile O at neutral pH. This phenomenon is observed in the catalytic mechanism of carbonic anhydrase, which catalyzes the reaction. CO2 plus H2O leftwards harpoon over rightwards harpoon HCO3 plus H plus. In the carbonic anhydrase enzyme, the bound Zn2 is present at the base of the 15 alpha D active site. In this enzyme, imidazole rings of 3 its residues occupy 3 coordination sites, and a fourth site is occupied by water molecule. The Zn2 plus ion facilitates the release of a proton by polarizing the water molecule generating O. The nucleophilic O ion attacks the substrate CO2, which is bound to the enzyme's catalytic site converting it to bicarbonate. Finally, the bicarbonate ion is released, and another water molecule binds to regenerate the catalytic site. Covalent catalysis. In covalent catalysis, the enhancement in the rate of reaction is brought through the formation of temporary covalent bonds amidst catalyst and substrate. The nucleophilic group on the enzyme reacts with an electrophilic group present on the substrate forming a covalent bond. The covalent bond formation among the enzyme and substrate helps in activating the substrate for specific reactions of the group enzyme belongs. The use of covalent modification as a catalytic strategy can be exemplified in the case of decarboxylation of acetoacetate catalyzed by primary amines. Initially, the cardinal group of acetoacetate is attacked by the nucleophilic amine yielding a shift base. The intermediate form has a protonated N atom acting as an electron sink which helps in stabilizing the transition state by reducing its enolate character. The synthesis and consumption of shift base occur rapidly, so this is not the rate determining step of the reaction. Enzyme kinetics. To understand the functioning of enzymes, the kinetics of enzymatic reactions needs to be described. In enzyme kinetics, we study the rate of the reaction and changes produced in response to experimental parameters. The number of moles of product produced per second is the rate of the catalytic reaction as designated by VO. The effect of increasing substrate concentration on the rate of catalysis varies in the presence of a constant concentration of enzyme, as depicted in figure 1.75. At first, the rate of reaction varies linearly with increasing substrate concentration. With further increase in substrate concentration, the increase in DO becomes smaller, starts to subside and ultimately approaches a maximum Vmax at larger concentrations of substrate. Figure 1.75 Michaelis Menten kinetics. The graph depicts the variation of reaction velocity VO with respect to the concentration of substrate. At much higher substrate concentration, Vmax approaches asymptotically. Km is the concentration of substrate where Rho is half of Vmax. Michaelis Menten equation. Adrian Brown examined the rate of hydrolysis of sucrose catalyzed by the enzyme beta-fructoronidase. Sucrose plus H2O right pointing arrow glucose plus fructose. He observed that when the concentration of sucrose is much greater compared to an enzyme, the rate of reaction becomes zero order for sucrose, i.e. rate becomes independent of sucrose concentration. Hence, Brown proposed that this reaction comprises two elementary reactions in which the substrate reacts with the enzyme to form a complex which subsequently breaks down to give product along with regenerating the. Hence, Brown proposed that this reaction comprises two elementary reactions in which the substrate reacts with the enzyme to form a complex which subsequently breaks down to give product along with regenerating the enzyme. E, S, and P represent the enzyme-substrate complex, and product, respectively. According to this equation, at a much higher concentration of substrate, when the whole of the enzyme has been converted into S complex, then the second step becomes the rate-limiting step of the reaction, and the rate of the reaction becomes independent of substrate concentration. In the equation, K1 and K-1 are the rate constants for forward and backward reactions leading to the formation of S complex, and K2 is the rate constant for conversion of S complex into enzyme and the product. Since the concentration of P is insignificant during the early stages of the reaction, it is assumed that the rate of reverse reaction designated by K-2, i.e. converting P back to S, can be neglected. This idea was further expanded by Leonor Michaelis and Mott Menten in 1913 into a general theory of enzyme action. The Michaelis-Menten equation illustrates the rate of the enzyme catalyzed reaction represented by equation 5 as a function of the concentration of substrate. This equation has been derived by hypothesis stating the breakdown of S complex to give the product, and the enzyme is the rate-limiting step of the kinetic reaction. The rate of the catalytic reaction is given by the product of rate constant K2 and concentration of S complex. The rate of reaction is given by the expression V0 equals K2 S6. Because S in equation 6 cannot be measured experimentally, we should express it in terms of alternative quantities. The rates of production and decomposition of S can be given by rate of production of S equals K1 E S7. Rate of decomposition of S equals K minus 1 plus K2 S8. Here, an assumption is made. Concentration of intermediate S remains constant until all the substrate has been used up. This is a steady state assumption, and hence, the rate of synthesis of S equals the rate of consumption of S throughout the reaction progress. K1 ES equals K minus 1 plus K2 S9. 1 to 1. S SKK. SK. Minus plus equals. The concentration of free enzyme E can be represented by total enzyme concentration at minus the concentration of S complex S. The concentration of substrate is usually very high as compared to total enzyme concentration, hence the quantity of substrate bound by the enzyme is insignificant compared to total substrate concentration. E equals ET S13. Substituting this expression for E into equation 12. TM. ESS. The above rate equation is for a single substrate enzyme catalyzed reaction called the Michaelis-Menten equation. This equation derives the relation between the initial rate reaction VO, the maximal reaction rate Vmax, and the initial concentration of substrate associated via Michaelis constant Km. When the substrate concentration S equals Km, then VO equals Vmax, 2 such that Km is the substrate concentration where VO is half of Vmax. The hypothesis used to simplify the derivation of the Michaelis-Menten equation limits its applicability to the initial aspects of the reactions where the concentration of enzyme is much shorter than the sum SO plus Km. For kinetic experiments, SO is much smaller than Km, hence for the equation to hold true, the condition is SO and GT EO. Determination of Vmax. The Vmax is determined by algebraic transformation of the Michaelis-Menten equation. A definite value of Vmax is difficult to obtain from plot of VO versus S, figure 1.75, as the value is reached asymptotically. However, an accurate value of Vmax can be obtained by deducing a linear form of Michaelis-Menten equation. By taking reciprocal of equation 18. MO max. KS1 volt VS. Plus equals. 19. M. O max max. SK1 volt VS VS. Equals plus. The enzymes which obey the Michaelis-Menten equation give a straight line when 1 VO is plotted against 1 S with an intercept of 1 Vmax, a slope of Km Vmax and the intercept on 1 S axis is minus 1 Km. This plot is also known as the double reciprocal plot. Determination of Km. The value of the kinetic parameter Km is also obtained from a double reciprocal plot of 1 VO against 1 S. The intercept on 1 S axis after extrapolation is minus 1 Km. Significance of Km. Km is the substrate concentration when VO is own half Vmax and thus expressed in concentration units. The Km values are independent of the concentration of enzyme and substrate. They provide information regarding the affinity of substrate for enzymes. For a given enzyme, various substrates react with it with different values of Km. Similarly, for a given substrate, different enzymes acting on it have different Km values. The Km value for an enzyme is affected by the specific substrate present, pH, temperature, and ionic strength. An enzyme having a small Km value shows maximum catalytic efficiency at lower substrate concentrations. When S and LT, Km, the reaction rate varies linearly with S VO equals Vmax S Km. When S and GT, Km, the enzyme is saturated VO equals Vmax. For the majority of enzymes, Km ranges from 10 to 1 and 10 to 7 M. Determination of KD. The relation between Km and rate constants of elementary reactions is depicted in the earlier scheme by equation 11. Km equals K minus 1 plus K2 K1. Assuming if K2 and LT, K minus 1, then enzyme substrate complex reverts to give E and S faster than the formation of product. Then equation 5.11 becomes 1 M 1 K KK. Therefore, under the conditions when K1 is much greater than K2, Km becomes equal to the dissociation constant KD of S complex. Under the circumstances when K2 and LT, K minus 1, Km becomes dissociation constant KD of the S complex. The KD values provide information on the affinity of enzymes for substrates in the S complex. Higher values of KD signify weak binding, and lower KD signifies a strong bonding. Determination of Kcat. We know that the maximum rate of reaction, Vmax, for an enzyme which undergoes two-step reaction is given by Vmax equals K2 ET. However, for enzymes undergoing multiple reaction steps, it is important to describe a rate constant, Kcat, which is equal to the rate constant for rate determining step of enzyme catalyzed reaction. This catalytic constant is also known as the turnover number of the enzyme, which is the number of substrate molecules catalyzed by each active site of the enzyme per unit time. The catalytic constant is expressed in reciprocal time units. Catalytic constants, Kcat is given by Kcat equals Vmax ET23. The parameters Kcat and Km are beneficial in evaluating the kinetic efficiency of enzymes. It is possible for two enzymes that catalyze different reactions to have the same value of Kcat or turnover number, but since the rate of uncatalyzed reactions can be different, so the rate acceleration caused by the enzymes can vary a lot. To compare the efficiencies of two enzymes, one should compare the ratio of Kcat Km for different reactions. When S and LT, Km, the equation 18 becomes. Hence, the rate of reaction depends on the concentration of E and S second order rate constant, or in other words, this implies that the rate depends on the encounter of the substrate and the enzyme in the solution. The values of Kcat Km for most of the enzymes lie near the diffusion control limit of 10 8 to 10 9. M minus 1 S minus 1. Since the decomposition of S complex to give product cannot takes place faster than its formation, hence the value of Kcat Km is never greater than K1. Lineweaver-Burke plot. For estimating the values of Km and Bmax, the Michaelis-Menten equation has been rearranged in several ways to obtain a linear relationship from an inaccurate fitting to the experimental data points. The plot, which is also known as the double reciprocal plot, can be obtained by rearranging the Michaelis-Menten equation 18 as 1 meter O max max K BBSV equals plus 27. The above equation reveals that plotting 1 BO against 1 S has a slope of Km Bmax. The intercept on 1 BO axis is 1 Bmax and the intercept on the 1 S axis is minus 1 Km. Figure 1.76. Figure 1.76 a line meter Burke plot. The graph is obtained by plotting 1 BO versus 1 S. The slope is a Km Bmax. The intercept is 1 Bmax 1 BO axis and the intercept is minus 1 Km on the 1 S axis. Using the line meter Burke plot, we can more precisely determine if Bmax is compared to the approximated value obtained from simply plotting BO versus S. In the reciprocal plot, the presence of less accurate data determined at low substrate concentrations leads to slope alterations of the line due to the long lever arm effect. Thus KCAD and KM values are overestimated. Nowadays, these parameters are obtained in a more efficient way by fitting the plot. Figure 1.77 of S BO versus S is linear, having a slope 1 Bmax. The intercept on the y-axis is Km Bmax and the intercept on x-axis is Km. Figure 1.78 Haynes-Wolff plot. The graph is obtained by plotting S BO versus S. The slope is given by 1 Bmax, intercept of Km Bmax on vertical axis and intercept of Km on horizontal axis. Enzyme inhibition. Competitive inhibition. A competitive inhibitor is one which is directly competing with the substrate for enzyme's active site. The binding of the inhibitor to enzyme's active site prevents the binding of the substrate with the enzyme. A competitive inhibitor usually resembles the substrate and combines with the enzyme to form a complex, but it does not lead to catalysis. This type of inhibitor slows down the catalysis rate by decreasing the concentration of free enzymes accessible for substrate binding. This type of inhibitor slows down the catalysis rate by decreasing the concentration of free enzymes accessible for substrate binding. The competitive inhibition can be represented by the given reaction scheme. Here is the inhibitor. A is a catalytically inactive enzyme inhibitor complex. An. Progress curves to the solution of differential or integrated rate equations is compared to initial velocity assay. Eddie-Hofstee plot. The Eddie-Hofstee plot is obtained by rearranging the Michaelis-Menten equation 18 as OOM max BBKB. The above equation reveals that the plot of BO against BOS is linear with a slope Km. The intercept on BO axis is Bmax and an intercept on BOS axis is Bmax Km. Though the Eddie-Hofstee plot generated relatively more reliable estimates, but due to the presence of dependent variable, B in both the axes makes error analysis more difficult. Figure 1.77 Eddie-Hofstee plot. The graph is obtained by plotting BO versus BOS. The slope is given by Km. The intercept of Bmax on the vertical axis and an intercept of Bmax Km on the horizontal axis. Haynes-Wolff plot. The Haynes-Wolff plot can be obtained by rearrangement of the Michaelis-Menten equation 18 as MO max max SSKBBB equals plus 29. Assumption is made that the binding of the inhibitor to the enzyme is reversible in nature and is in equilibrium with it. The dissociation constant for the inhibitor is I EI K A equals 30. In the presence of a competitive inhibitor, the Michaelis-Menten equation can be derived as before, but with an additional term to account for a fraction of ET that binds to I forming AIE. ET equals E plus S plus A31. The equation becomes max O M BS. Function of the concentration of the inhibitor and its affinity for the enzyme. The presence of I makes Km appear to be larger than it really is and makes S appears to be less than it really is. Since I bind to the enzyme reversibly, hence by increasing the substrate concentration, the effect of competitive inhibition can be reversed. The substrate concentration needs to be increased by factor of alpha to reverse the effects of the presence of an inhibitor. As the S far exceeds I, BO approaches Bmax for any value of alpha. The presence of a competitive inhibitor does not affect the Bmax value of the enzyme. Equation 32 can be rearranged in double reciprocal form. 111 MO max max K. The plot of the above equation is linear, having a slope of alpha Km Bmax, an intercept of minus 1 alpha Km on 1 S axis, and an intercept of 1 Bmax on 1 BO axis. In the presence of a competitive inhibitor, the double reciprocal plots at varying concentrations of I intersect at 1 Bmax on the 1 BO axis, which is a characteristic of competitive inhibition. The value of alpha can be evaluated by the difference in slope at a given concentration by having the values of I and alpha. Ki can be obtained using equation 33. Figure 1.78 competitive inhibition. Double reciprocal plot of enzyme kinetics in the absence and presence of competitive inhibitor. Competitive inhibitor increases Km but does not affect Bmax. Non-competitive inhibition. A non-competitive inhibitor shows equal binding affinity for both the enzyme and the enzyme substrate complex. Non-competitive inhibition is viewed as special case of mixed inhibition where Ki is equal to Ki. The dissociation constant for inhibitor binding steps is I. The Michaelis-Menten equation in the presence of a non-competitive inhibitor can be derived as before but with additional terms to account for a fraction of ET that binds to E and S, forming A and ESI, respectively, i.e. ET equals E plus S plus A plus ESI. In the presence of a non-competitive inhibitor, the Bmax value is lowered and the value of Km remains unaffected. We can say that the inhibitor lowers the concentration of functional enzymes. The corresponding equation for the double reciprocal plot is given by the double reciprocal plots for non-competitive inhibition contain lines having slope alpha Km Bmax, an intercept of alpha Bmax on 1 BO axis and an intercept of minus 1 Km on 1 S axis. These linear plots for a non-competitive inhibitor at different concentrations of I converge to the left of 1 BO axis at minus 1 Km. Increasing the substrate concentration cannot reverse the non-competitive inhibition. It has been observed that this type of enzyme inhibition is mainly found in textbooks and rarely in real experimental data. The reason for this apparent paradox is ascribed to the assumption that free substrate and substrate bound to the enzyme are in equilibrium. Figure 1.79 non-competitive inhibition. A double reciprocal plot of enzyme kinetics in the absence and presence of non-competitive inhibitor. The presence of non-competitive inhibitor decreases the Bmax value and does not affect the value of Km. Incompetitive inhibition. An incompetitive inhibitor is the one that binds to the enzyme substrate complex but shows no binding on the enzyme's active site. This type of inhibitor only affects the catalytic activity of the enzyme and does not affect its substrate binding. The incompetitive inhibition can be described by following the reaction scheme. The dissociation constant K1 for the inhibitor binding step is given by I SI. The Michaelis-Menten equation in the presence of an incompetitive inhibitor can be derived as before but with an additional term to account for a fraction of ET that binds to S forming ESI, i.e. ET equals E plus S plus ESI. The equation the double reciprocal plots give a series of parallel with slope Km Bmax, intercept of alpha, Bmax on 1 BO axis, and intercept of alpha, Km on 1 S axis. Thus, the presence of incompetitive inhibitor affects only intercepts of the double reciprocal plots, and slopes remain unaffected. Since an incompetitive inhibitor shows no binding to the active site of the enzyme, hence its effect cannot be undone by increasing the substrate concentration. The incompetitive inhibition is commonly observed for multi-substrate enzymes. Figure 1.80 incompetitive inhibition. A double reciprocal plot of enzyme kinetics in the absence and presence of an incompetitive inhibitor. The presence of incompetitive inhibitor affects the value of both Km and Bmax. Mixed inhibition. Mixed inhibition can be described as a combination of two different types of inhibition, competitive inhibition and incompetitive inhibition. Many inhibitors react with the enzyme and alter their substrate binding and catalytic activity. The binding of mixed inhibitors takes place at sites of the enzyme, which takes part in substrate binding as well as catalysis. The reaction model is given by the two dissociation constants for inhibitor binding are not equivalent. I. In contrast to non-competitive inhibition, both Km and Bmax are affected in the presence of mixed inhibitors. The double reciprocal plots for mixed inhibition consist of lines having slope alpha km Vmax, and an intercept of alpha, Vmax on one BO axis, and an intercept of alpha km on one S axis. The lines for increasing values of I representing increasing saturation of E and S with I intersect on the left side of one BO axis. Figure 1.81 mixed inhibition. A double reciprocal plot of enzyme kinetics in the absence and presence of mixed inhibitors. Both Km and Vmax are affected by the presence of mixed inhibitor. Feedback inhibition. To conserve the building blocks and metabolic energy, the enzymatic activities need to be controlled. In a multi-enzyme system, if the concentration of the end product surpasses the necessity of the cell, it leads to inhibition of enzyme catalysis in the committed first step of the pathway. Slowing down the first enzyme results in the diminished activity of the subsequent enzymes due to depletion of their substrates. Ultimately, the amount of end product synthesized is balanced according to the needs of the cell. This kind of inhibition is known as feedback inhibition. Feedback inhibition can be exemplified in the case of a bacterial enzyme system catalyzing the transformation of L-threonine to L-isoleucine in 5 steps. In this pathway, the end product of the sequence, isoleucine, is found to inhibit the enzyme threonine dehydratase catalyzing the first step of the sequence. Isoleucine is shown to exhibit remarkable specificity as an inhibitor because it inhibits only threonine dehydratase in the series. The isoleucine inhibits the enzyme by binding at a regulatory site which is different from the active. The isoleucine inhibits the enzyme by binding at a regulatory site which is different from the active site of the enzyme. The isoleucine inhibits the enzyme by binding at a regulatory site which is different from the active site of the enzyme. This binding of isoleucine is reversible and non-covalent. Thus, the enzyme threonine dehydratase exhibits decreased or increased catalytic activity in response to variation in the concentration of isoleucine. Figure 1.82 feedback inhibition. A series of 5 enzymes E1 to E5 catalyzes the formation of L-isoleucine from L-threonine. The end product of the sequence, L-isoleucine, allosterically inhibits the enzyme E1, threonine dehydratase. The enzyme is specific for L-isoleucine as an inhibitor as it is not inhibited in any other intermediate of the sequence. Glycolysis. Overview of glycolysis. Glucose is a major source of energy in most cells and tissues. It also acts as a versatile precursor, capable of supplying a huge array of intermediates for biosynthetic reactions. Glucose has 3 metabolic fates in most of the organisms, animals, and plants. It is stored in the form of polysaccharides as starch or glycogen. Pyruvate is oxidized to create ATP and metabolic intermediates via glycolysis. Ribose 5-phosphate nucleic acid production, and NADH is oxidized via the pentose phosphate pathway, reductive biosynthetic processes. Glucose plus 2 ADP plus $2 million plus 2 pi right pointing arrow 2 pyruvate plus 2 ATP plus 2 NADH plus 2 H plus. Glycolysis is anaerobic as it does not require oxygen but can occur in the presence of oxygen as well. However, pyruvate can be further processed either aerobically to CO2 and H2O, oxidative phosphorylation, or anaerobically to lactic acid fermentation, or ethanol alcoholic fermentation. Figure 1.83 possible metabolic fates of glucose. 3 important fates of glucose: storage, oxidation via glycolysis, and oxidation via pentose phosphate pathway. Glycolysis is divided into 2 phases and comprised of 10 enzyme catalyzed reactions. The first 5 steps constitute the preparatory phase or investment phase, where the energy of ATP is invested, and the 6-carbon compound gets converted into the common product glyceraldehyde 3-phosphate, and the latter 5 into the payoff phase or harvesting phase, where energy is conserved in the form 2 ATP and 2 NADH molecules per glucose molecule. The first phase of glycolysis, preparatory phase. In this phase of glycolysis, 2 molecules of ATP are consumed, and the hexose sugar is cleaved into 2 trisomoieties. Step 1. Phosphorylation of glucose hexokinase. In the first step, the conversion of glucose to glucose minus 6-phosphate is catalyzed by hexokinase. Hexokinase is an enzyme that phosphorylates a hexose to a hexose phosphate. There are several hexokinase isoforms or isozymes having different functions in the same organism, viz. Hexokinase I, housekeeping enzyme, hexokinase 2, hexokinase 3, and hexokinase IB, popularly known as glucokinase. Figure 1.84 2 phases of glycolysis, preparatory phase. Phosphorylation of glucose and conversion to glyceraldehyde 3-phosphate, payoff phase. Conversion of glyceraldehyde 3-phosphate to pyruvate with the concomitant formation of ATP and NADH. For each molecule of glucose, 2 molecules of pyruvate are formed and 2 molecules of ATP and 2 molecules of NADH. Step 2. Conversion of glucose 6-phosphate to fructose 6-phosphate. Phosphoglucose isomerase. Phosphoglucose isomerase is involved in the interconversion of glucose 6-phosphate and fructose 6-phosphate. This reaction is driven by the relative concentrations of these sugars in the cytoplasmic matrix of the cell. Phosphoglucose isomerase is a multifunctional enzyme that functions as neurolutin, autocrine motility factor, differentiation and maturation mediator, and is a myofibril-bound serine protease inhibitor. Therefore, it has functions in glucose metabolism inside the cell, while outside the cell, it acts as a nerve growth factor and cytokine. Mutations in phosphoglucose isomerase are associated with non-spherocytic hemolytic anemia. Step 3. Phosphorylation of fructose 6-phosphate to fructose 1,6-phosphate phosphofructokinase. This is the second kinase reaction of the pathway catalyzed by phosphofructokinase 1, commonly referred to as phosphofructokinase. Phosphofructokinase 1 PFK1. Phosphofructokinase deficiency leads to Tarui disease, glycogen storage disease 7, an autosomal recessive disorder characterized by severe nausea, vomiting, muscle cramps, and myoglobin in response to bursts of intense or vigorous exercise. Step 4. Cleavage of fructose. Phosphofructokinase deficiency leads to Tarui disease, glycogen storage disease 7, an autosomal recessive disorder characterized by severe nausea, vomiting, muscle cramps, and myoglobin in response to bursts of intense or vigorous exercise. Step 4. Cleavage of fructose 1,6-bisphosphate aldolase. In this step, fructose 1,6-bisphosphate is split into 2 3-carbon molecules by the enzyme fructose-bisphosphate aldolase aldolase. One of these is an aldose, dihydroxyacetone phosphate, and the other is ketose, glyceraldehyde 3-phosphate. This aldol condensation is readily reversible under intercellular conditions. Step 5. Interconversion of triphosphate. The enzyme triphosphate isomerase rapidly and reversibly converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. The second phase of glycolysis, payoff phase. In the payoff phase of glycolysis, energy is conserved in the form of ATP and NADH. Step 6. Conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate glyceraldehyde 3-phosphate dehydrogenase. The first step of the payoff phase is the reversible oxidation of glyceraldehyde 3-phosphate to a high-energy acyl derivative 1,3-bisphosphoglycerate catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Step 7. Phosphoryl transfer from 1,3-bisphosphoglycerate to ATP. Phosphoglycerate kinase. Phosphoglycerate kinase transfers a phosphate group from the acyl phosphate group of 1,3-bisphosphoglycerate to ATP generating ATP, i.e., substrate level phosphorylation, and 3-phosphoglycerate. Step 8. Conversion of 3-phosphoglycerate to 2-phosphoglycerate phosphoglycerate mutase. This reaction involves the rearrangement of the phosphate group on the carbon 3 C3 of 3-phosphoglycerate to carbon 2 C2 resulting in the formation of 2-phosphoglycerate catalyzed by phosphoglycerate mutase. Step 9. Dehydration of 2-phosphoglycerate to phosphoenol pyruvate analase. The enzyme analase catalyst is the reversible removal of water molecule from 2-phosphoglycerate to generate phosphoenol pyruvate, a compound with high transfer potential of the phosphoryl group. Step 10. Transfer of the phosphoryl group from phosphoenol pyruvate to ADP pyruvate kinase. In the final step, the enzyme pyruvate kinase catalyst is the transfer of phosphate group from the phosphoenol pyruvate to ADP generating ATP and pyruvate. This is the second substrate level phosphorylation reaction of the pathway, and pyruvate kinase requires both K+ and Mg2+ or Mn2+. Pyruvate kinase deficiency causes hemolytic anemia, where it is unable to form ATP resulting in cell damage. Even cells become swollen and are taken up by the spleen, causing splenomegaly. Other signs and symptoms include jaundice, icterus, and elevated bilirubin. Arsenic poisoning also prevents ATP synthesis, as it occupies the place of phosphate in steps of glycolysis. Serum pyruvate kinase M2 isoform level is higher in cancer cells as compared to healthy cells. Hence, serum pyruvate kinase M2 levels are used as markers for cancer. Thus, the overall balance sheet will show the net gain of ATP. Hence, 1 molecule of glucose on complete conversion by glycolytic pathway results in the yield of 2 pyruvate molecules, 2 NADH and 2 ATP molecules, table 1.13. Step reaction type enzyme. Delta G, degree in kJ. Mol -1. 1. Glucose plus ATP right pointing arrow glucose 6-phosphate plus ADP plus H plus. Phosphoryl transfer. Hexokinase -16.7. 2. Glucose 6-phosphate rightwards arrow over leftwards arrow fructose 6-phosphate. Isomerization phosphoglucose isomerase. Plus 1.7. 3. Fructose 6-phosphate plus ATP right pointing arrow fructose 1. 6-bisphosphate plus ADP plus H plus. Phosphoryl transfer. Phosphofructokinase. Minus 14.2. 4. Fructose 1. 6-bisphosphate rightward 0 over leftward 0 dihydroxyacetone phosphate plus glyceraldehyde 3-phosphate aldyl cleavage aldolase plus 23.8. 5. Dihydroxyacetone phosphate rightward 0 over leftward 0 glyceraldehyde 3-phosphate isomerization triphosphate isomerase. Plus 7.5. 6. Glyceraldehyde 3-phosphate plus pi plus NAD plus rightward 0 over leftward 0 1. 3-bisphosphoglycerate plus NADH plus H plus. Phosphorylation coupled. 2 oxidation glyceraldehyde 3-phosphate dehydrogenase plus 6.3. 7. 1. 3-bisphosphoglycerate plus ADP rightward 0 over leftward 0 3-phosphoglycerate plus ATP phosphoryl transfer. Phosphoglycerate kinase. Minus 18.8. 8. 3-phosphoglycerate rightward 0 over leftward 0 2-phosphoglycerate phosphoryl shift phosphoglycerate mutase. Plus 4.6. 9. 2-phosphoglycerate rightward 0 over leftward 0 phosphoenol pyruvate plus H2O dehydration enolase plus 1.7. 10. Phosphoenol pyruvate plus ADP plus H plus right pointing arrow pyruvate plus ATP. Phosphoryl transfer. Pyruvate kinase minus 31.4. Note, delta G, degree is the standard free energy change. Table 1.1 reactions of glycolysis with a standard free energy change at each step in erythrocytes. The metabolic phase of NADH and pyruvate. The products of glycolysis cells have to maintain the redox balance, and also it is critical that NADH is recycled back to NAD+ so that the glycolysis proceeds. Without NAD+, the payoff phase will halt and cause a backup in glycolysis. NAD+ is regenerated by one of the three catabolic pathways for pyruvate metabolism. One under aerobic conditions, oxidation by citric acid cycle, and the other two in anaerobic conditions are lactic acid fermentation and alcohol fermentation. Figure 1.85 Three catabolic phase of pyruvate. Pyruvate is the end product of glycolysis, which under aerobic conditions gets oxidized to CO2 and H2O, and under anaerobic conditions to ethanol or lactate. Regulation of the glycolysis. Intermediates are generated and consumed at the same rate in a metabolically active cell in a steady state. During perturbations or variations in the rate of creation or consumption of a molecule, compensating adjustments in enzyme activities bring the system to a steady state. Also, the regulatory mechanisms maintain the nearly constant levels of key metabolites such as ATP, NADH, and glucose in the blood. Thus, the flux through the glycolytic pathway must be adjusted in response to intracellular and extracellular conditions. There are 3 steps in glycolysis corresponding to the steps with the largest negative free energy changes which regulate the flux of glycolysis. Reaction 1 hexokinase, reaction 3 phosphofructokinase, and reaction 10 pyruvate kinase. Some of the enzymes of glycolysis and gluconeogenesis are also regulated by post-translation modifications such as acetylation and alkylation. Figure 1.86 Standard free energy changes during the reactions of glycolysis. Regulation of glycolysis occurs at the steps where the largest negative free energy change and irreversible reactions of the pathway reactions 1, 3, and 10. Regulation of hexokinase. 4 isozymes of hexokinase I, 2, 3, and IB are present in humans. The combined action of blood 4 on plasma membrane and hexokinase I3 in the cytosol maintains a balance between glucose uptake and phosphorylation in skeletal muscles. Hexokinase catalysis is the first step in glycolysis and is allosterically inhibited by its product glucose 6-phosphate. If glucose 6-phosphate accumulates in the cell, there is feedback inhibition of hexokinase till it is consumed. Hexokinase I, 2, 3 is not regulated by any of the downstream intermediates or products of metabolism. Figure 1.87 Comparison of the kinetic activities of hexokinases I, IB. Hexokinases other than glucokinase have low Km for glucose. This difference accounts for the rapid conversion of glucose to glucose 6-phosphate in non-hepatic tissues and the delivery of glucose to the blood by the liver. Figure 1.88 Regulation of glucokinase. Reversible binding of GKRP, glucokinase regulatory protein, inhibits the activity of glucokinase, and this inhibition is overcome by fructose 1-phosphate. The binding of GKRP to glucokinase is also enhanced by fructose 6-phosphate. Regulation of phosphofructokinase. The irreversible metabolic reaction catalyzed by phosphofructokinase is the most important regulatory step of the glycolytic pathway, which commits glucose towards glycolysis. In addition to substrate binding sites, this enzyme has several regulatory sites, where allosteric activators or inhibitors bind. The enzyme activity is controlled by complex allosteric regulation. ATP is the end product of glycolysis as well as a substrate for phosphofructokinase. ATP binds to the inhibitory site of the enzyme at a high concentration of ATP and thereby decreasing the activity of the enzyme and affinity for the fructose 6-phosphate. Allosteric activation of the enzyme caused by the increased concentration of ADP or AMP overcomes the inhibitory effect of ATP. High concentrations of citrate, an intermediate of the aerobic oxidation of pyruvate, fatty acids and amino acids, enhance the inhibitory effect of ATP for phosphofructokinase, further reducing the ethyl glucose in glycolysis. Fructose 2, 6-bisphosphate is a major allosteric activator of phosphofructokinase. At low blood glucose levels, glucagon signals the liver to produce and release more glucose rather than using it for its own need. Fructose 2, 6-bisphosphate activates the phosphofructokinase by increasing its affinity for fructose 6-phosphate and diminishes the inhibitory effect of ATP and citrate. This is an example of feed-forward stimulation as glycolysis is accelerated when glucose is abundant. Figure 1.89 Regulation of phosphofructokinase and fructose 1, 6-bisphosphate. Fructose 2, 6-bisphosphate is a major allosteric activator of phosphofructokinase, and its high level activates the phosphofructokinase, while a low level increases the activity of fructose 1, 6-bisphosphate. Regulation of pyruvate kinase. The enzyme pyruvate kinase catalysis is the third irreversible step in glycolysis, which controls the outflow from this pathway. Several isozymes of pyruvate kinase encoded by different genes are found in vertebrates, differing in their tissue distribution and response to modulators. The L-type predominates in the liver, while the M-type is predominant in the brain and muscle. Fetal erythrocytes have low concentrations of glycolytic intermediates as compared to adults due to high activity of pyruvate kinase. Due to the low steady-state concentration of 1, 3-bisphosphoglycerate, the shunt of 2, 3-bisphosphoglycerate is largely reduced in fetal cells resulting in less formation of 2, 3-bisphosphoglycerate. This is to ensure the oxygen supply to fetal cells, as 2, 3-bisphosphoglycerate is a negative regulator of hemoglobin affinity for oxygen. Pyruvate kinase activity is inhibited under low glucose conditions by covalent phosphorylation, which differs among the different isozymes. Figure 1.90 Regulation of pyruvate kinase. Most animals for maintaining blood glucose levels from dropping too low hypoglycemia. The other means of maintenance is through the degradation of glycogen glycogenolysis. Gluconeogenesis occurs during fasting, starvation, low carbohydrate diets, or intense exercise and usually takes place in the liver. This pathway consists of 11 enzyme catalyzed reactions, of which most of the reactions are the reversible steps found in glycolysis. The amounts and the activities of these essential enzymes also are regulated. Fructose 1, 6-bisphosphate, AMP and ADP act as a positive modulator of the enzyme. ATP, acetyl-CoA, and long-chain fatty acids allosterically inhibit all the isozymes of pyruvate kinase. Connection between regulation of glycolysis and gluconeogenesis. Gluconeogenesis is a metabolic pathway for the production of glucose from non-carbohydrate carbon substrates, lactate, intermediates of citric acid cycle, amino acids, and glycerol. Gluconeogenesis is one of the 2 main mechanisms adopted by humans and Figure 1.91 Reciprocal regulation of gluconeogenesis and glycolysis in the liver. Level of fructose 2, 6-bisphosphate, ATP and ADP, and phosphorylation of pyruvate regulate the activities of the enzymes of both glycolytic and gluconeogenic pathways. Coordination between glycolysis and pentose phosphate pathway. Glucose 6-phosphate is metabolized by both the glycolytic pathway and the pentose phosphate pathway (PPP), and its metabolism by PPP is coordinated with glycolysis. The cytoplasmic concentration of NADP+ plays a key role in determining the fate of glucose 6-phosphate. The first reaction is the dehydrogenation of glucose 6-phosphate in the oxidative branch of the PPP. This is an irreversible reaction and a rate-limiting step under physiological conditions. The marked effect of the NADP+ level on the rate of the oxidative phase ensures that NADPH generation is tightly linked to its utilization in reductive biosynthesis. The reductive phase of the PPP is controlled primarily by the availability of substrates. The understanding of the intricate interplay between glycolysis and the PPP could be done by examining the metabolism of glucose 6-phosphate in 4 different metabolic situations. Figure 1.92. Figure 1.92 different modes or situations of glucose 6-phosphate metabolism. Mode 1 ribose 5-phosphate is in demand. Mode 2 balance requirement of NADPH and ribose 5-phosphate. Mode 3 NADPH is in demand and mode 4 ATP and NADP are in demand. Utilization of other substrates in glycolysis. Glycolysis is both principal route for glucose metabolism and the main pathway for the metabolism of fructose, galactose, and other dietary carbohydrates. They enter the pathway after being transformed into one of the glycolytic intermediates. Figure 1.93. Figure 1.93 feeder pathways for glycolysis. The bridging step oxidative decarboxylation of pyruvate. Although the TCA cycle begins with acetyl CoA that condenses with oxaloacetate to form carboxylate citrate, we first need to briefly start our discussion with the formation of pyruvate, the 3-carbon end product of glycolysis. Glycolysis occurs in the cytosol of both prokaryotes and eukaryotes, and it is thought to be the best understood pathway to date. Through glycolysis, each molecule of glucose undergoes successive reactions catalyzed in 10 steps with the generation of 2 molecules of pyruvate, 2 NADH, 4 ATPs, net 2 ATPs, and 2 H2O, as diagrammatically shown. Glycolysis is discussed in detail in the glycolysis chapter. Substrates other than glucose, both simple and complex, can be utilized via glycolysis after enzymatic conversion to one of the glycolytic intermediates. The citric acid cycle. The citric acid cycle, alternatively known as the tricarboxylic acid TCA cycle in Krebs cycle after its discovery by Hans Krebs, carries out the majority of oxidation of the carbohydrate, amino acid and fatty acid, as well as produces a myriad of biosynthetic precursors to be used in the synthesis of molecules of immense cellular importance. TCA cycle is amphibolic as it operates anabolically and catabolically in both prokaryotes and eukaryotes. Figure 1.94 diagrammatic representation of glycolytic steps. Each cycle of glycolysis undergoes 10 successive steps, catalyzing the breakdown of one molecule of glucose to 2 molecules of pyruvate, or we can say the complete glycolytic breakdown of 1 mole of glucose, 6.023 times 1023, yields 2 moles of pyruvate, 12.046 times 1023, in the cell with the generation of 2 NADH, 4 ATPs, net 2 ATPs, and 2 H2O. Entry of pyruvate in mitochondria. Mitochondrion is a semi-autonomous, double membrane bound organelle located in the others. In the matrix of each mitochondrion, there are several copies of covalently closed circular cccDNA called mtDNA, numerous